For neurons to communicate, vesicles containing neurotransmitters must fuse with the plasma membrane at the axon terminal. They do this with the help of 6 proteins: syntaxin 1, MUNS18, MUNS13, SNAP25, synaptobrevin, and synaptotagmin. Syntaxin 1 lives in the plasma membrane of the axon terminal, usually in complex with MUNS18. With MUNS18 bound to it, syntaxin 1 stays in a closed conformation, unable to bind to other proteins. When MUNS13 enters and associates with syntaxin 1, it causes a conformational change that will expose a region on syntaxin 1 called the snare domain, which is key for binding the other proteins involved in membrane fusion. Soluble SNAP25 can then bind to the exposed snare domain and form a partial snare complex with syntaxin 1. When a vesicle approaches a partially formed snare complex, a protein in the vesicle membrane called synaptobrevin can bind to the snare complex. This will bring the vesicle and plasma membrane into close association, but not close enough to fuse. This is because another vesicle protein called synaptotagmin also binds at the same time, preventing synaptobrevin from fully zippering with the snare complex. But once the action potential reaches the axon terminal, the influx of calcium ions causes a conformational change in synaptotagmin, allowing the snare complex to completely zipper. This creates enough bend and tension for the 2 membranes to fuse, and the neurotransmitter is released into the synaptic cleft to send the signal on to the postsynaptic neuron.